We've been saying for decades that dinosaurs were on the ark, and they certainly could have fit, no problem. Even these sauropods like we've got here. By the way, I've had this since like the 1970s. This is my brachiosaur toy, and it was featured in Jurassic Park 1993.
The first one, yeah.
Incorrectly, but I thought, I'll just bring it as a homage. So even those would have gone on the ark, but they don't have to be the giant grandparent versions. They're just the juvenile ones.
Hello, everyone. Welcome to the Creation Podcast, the show where we discuss the science that confirms Scripture. I'm your host, Trey. In 1993, the movie Jurassic Park took the world by storm with its compelling characters, excellent cinematography, industry-altering special effects, and unfortunately faulty science. While dinosaurs had been popular, this modern classic of a film rocketed these reptilian beasts to a new level of stardom. And while the film's sequels have been hit or miss, it's difficult to overstate the effect the franchise has had on public perception of dinosaurs. The question remains, is building a Jurassic Park possible? Common sense tells us no. Even more common sense says there's been a whole movie franchise dedicated to telling us that it would be a bad idea to even attempt something like this. But what does the science say? How possible is it really? And to discuss this topic with me today, I have ICR's paleobiochemist, Dr. Brian Thomas. Thank you so much for being here, Dr. Thomas.
Well, of course, it's my pleasure.
Absolutely. I know you love dinosaurs. I know that Jurassic Park features some things that are in your particular wheelhouse of expertise. And so this, I anticipate, will just be a fantastic conversation. Of course, I think everyone kind of already knows we're not going to build a Jurassic Park, but let's kind of discuss what is possible, what isn't possible, what we can know, what we can't know, based on what we see in that movie and what the science actually says. So, to start,
we see a lot of fiction.
We do see a lot of fiction. We see a lot of fiction.
So we'll just lay that out there right at the beginning.
A lot of fiction. It is a good story. It's a good story, but that doesn't mean that it lines up with reality. So, Jurassic Park features the use of DNA found in a mosquito trapped in amber. I mean, this is the inciting incident. They find this mosquito in amber that's supposedly millions of years old. And then in that mosquito, they find some leftover remnants of the DNA. According to just standard old Earth ideologies, how old would it make that DNA in a mosquito like that?
70 million years, something like that. So when we excavate Cretaceous, which we call middle of the flood layers, we find dinosaur bones. In fact, I brought one with me, but it's bone fragment. So this is a dinosaur bone fragment. This is what we're talking about. And right alongside, you see little carbonized, turned into colified sticks, pieces of ancient plants. And next to those, in some of those sticks, you see little bitty drops of amber, and they're called resinites. So anyway, we've seen all these. So the idea would be, you just pull out a piece of amber, find a mosquito, pull the DNA out of its abdomen, and hopefully you're getting its last blood meal, and hopefully its last blood meal was a dinosaur, which I don't think it would have been, honestly, but because we know a lot about dino skin, and it was thick, super thick. So anyway, armor-plated skin and things like that. Anyway, those rocks and those fossils that are in them, whatever they may be, have an age assignment on the order of tens of millions of years. Of course, the Jurassic layers that we excavate have an age assignment of over, 100 million, something like that. So 70, 100, just unimaginable eons.
I guess Cretaceous Park didn't have the right ring to it.
That's all it was.
Yeah, they went for the best marketing name. Yeah, awesome.
Cinemanian Park. Wait, what?
Okay, so the question is then, given the events in the movie as it's presented, could DNA last 70 to 100 million years inside the abdomen of a mosquito from that long ago?
No, not at all.
Not at all.
Not at all. Yeah. So we have actually a great paper came out just, I guess it's been a decade or more ago now, but 2012. Mm-hmm. And the title of the paper is, and when I say paper, I mean technical science peer-reviewed journal paper.
Right.
The half-life of DNA in bone, measuring decay kinetics in 158 dated fossils. And this is published in Proceedings of the Royal Society B.
Not as marketable as Jurassic Park.
Not as marketable, but more interesting to me because this tells me this is like the best case scenario. Best case scenario, how long does DNA last? And they calibrated the decay rate of their DNA that they sequenced in Moa bones from New Zealand. Moa is an extinct giant bird. And so they got 158 Moa bones, and they sequenced the DNA, and they compared, how much DNA is in this Moa bone versus what's its carbon age. It's in carbon years, which, by the way, don't necessarily correspond to calendar years. It's a whole separate topic. But carbon years are older in many cases than calendar years, and I think older in most of these cases as well. So that means that when they calibrate the DNA to the carbon years, that means their DNA expectation of how long DNA can last is actually way longer than what it really realistically would be. But that's okay, because even with this fudge factor in favor of it lasting a lot longer, we get DNA decaying in thousands of years, not 1 million, not even close. So based on the decay curve that they produced, I wrote a news article and did some study. I did actually a technical journal article as well about a related issue. And I ran that curve out to see, okay, how many years out, according to this, how many theoretical years, according to the decay curve, would you have to run this in order for you to have no two DNA bases left linked together? So as we know, DNA is made of chemical bases, and they are covalently bonded, one base to another, in a long string, and that's what makes your DNA. And so, so what's happening underground? Whether it's in amber. And by the way, amber is not some sort of hermetic seal. Right. It's not a magic time capsule. That's what a lot of people perceive it as. It's like, well, you can preserve anything as long as it's in amber. Not at all. Amber develops cracks as it ages. It also darkens, it oxidizes, and it shrinks as well. It develops cracks into which air can penetrate. And so amber actually itself looks young. Just because it's when we find it in these deposits, it's not yet cracked. It's not yet dark sometimes. And so that's a separate topic. But okay, so here's what I wrote here. To have zero backbone bonds. Now those are the bonds that make the backbone of DNA, the bonds between nucleotides there. To have zero backbone bonds after approximately 350,000 years at 10 degrees Celsius. So the decay rate depends on temperature. And what do you think? The faster, you can make it go faster, decay faster, if you just add temperature and heat it up. And that means your molecules are bouncing around that much faster. And therefore the degradative or destructive chemistry that naturally and relentlessly and inevitably happens even faster at high temperature. So at 10 degrees Celsius, which is, I don't know the conversion of that, but 40 something maybe Fahrenheit. Maybe we could put a lower third with the actual.
Right. For those of you who don't want to do the math in your head, right. Here's in Fahrenheit.
Yeah.
Freedom units, right?
Is that what that is? Okay, so we're talking about, 350,000 years. That's max extension.
Right.
This is for various reasons. So anyway, the bottom line being the longest we can get this to last is less than 500,000 years for sure.
Okay. And that's best case scenario, meaning that it would typically deteriorate much faster than that.
Yeah, well, if you add more water, or if you change the pH, or if you change the salinity, or if you add bacteria, or the extra radiation, there's so many things that break DNA, that break down DNA. And so when DNA interacts with the world's natural environment, it is relentlessly destructive to DNA. And that's why in labs we always have to preserve our DNA in liquid nitrogen. We put it in a buffer to make it happy, and then we put that in liquid nitrogen for long-term storage. Because the colder it is, as we said, the less the molecules bounce around, and the longer we can get it to last.
Okay.
But there's no place on Earth except in these labs.
Very controlled environment.
Except where you have liquid nitrogen in production facilities or whatever you have liquid nitrogen. That's the only place that it stays that cold on Earth. So it's not a reasonable Earth surface temperature to expect DNA to. But we do have permafrost, and so these are places on Earth near the latitude, the polar latitudes, where the ground is always frozen, permafrost, permanently frozen. And there are fossils in there, and those actually have DNA in them, but there are no dinosaurs that we know of yet. So.
Gotcha. Okay, well, then the question is, have we found DNA in dinosaur? Have we found dinosaur DNA in this day and age?
This is depends on what you mean by DNA.
Okay.
Yeah. So we found it actually just in 2020, and then I think there's a paper in 2017. So there may be 3 reports. There were some initial reports in the early 1990s of dinosaur DNA, and that was all scoffed at. I mean, lampooned. Is that the word? Lampooned?
Lambasted?
Lambasted, maybe? Yeah, one of those lamb words. And because of the decay rate. And even back then, we had a good, solid idea of the decay rate of DNA, and we knew it couldn't last 1 million years at all. And so to find it or to report it, in dinosaur bone was just it's just unthinkable,
right? Because
we quote unquote know that bone is 70 million years old, so we can't be dinosaur DNA. So that's the logic that was used back then. So, but that is still, it did get published back then by Morrell, I think is his name. And anyway, so no one has done yet a sequence. No one has published a sequence of bases like A's, T's, G's, and C's that they extracted from the bone, and that does not match contamination sequence like bacteria or fungus. These are microbes that you would expect to be in there. So you have to have a long enough sequence to compare it with sequences that we do know of these potential microbe contaminants, and say, it doesn't match anything that these microbes have. Therefore, it's more likely to have come from the original bone. But anyway, it's just tough because what paleontologist, is going to put their reputation at risk by even looking for DNA sequence when everyone knows that DNA can't last as long as these are supposed to have been in there, in the ground. And so they haven't been looking on purpose because they don't want to find it. Because if they find it, then the same thing that happened to Morrell will happen to them, and they'll be booted out of the club and they won't be able to publish anymore. And that has happened, But they've gotten close as publishing, like I said, in 2020, I believe, and more recently, what looks like chromosomes. So chromosomes, it's DNA all packaged and wadded up and ready for cell division. Mm-hmm. That's what happens when we have chromosomes. And then after the cells divide, those chromosomes get unpackaged and the regular daily life of the cell commences. And then we call that chromatin because it's not all in its packaged form. And so, but when it's packaged in the form of chromosomes, it can take up DNA-specific stains. There's multiple of these stains. And they've found that in Cretaceous bird fossil, for example. and they found it in other fossils. I don't know about dinosaur, but certainly fossil cell. They find the cells that are still in there, and in these cells there are nuclei that's still in there. And in some of these nuclei, they throw the stain on there, and it absorbs the stain as though it's actually DNA. And the reason the stain is DNA specific is because it's a tiny little molecule that's like a little disc shape, and it fits right in between each of the rungs of the DNA ladder, if you will, molecularly. Think really tiny. It's like, little wafers that get stuck in between, in between the rungs of a ladder. So it picks up the stain. So it's one of these things where it's like, have they found dino DNA? Well, they found, what else could it be?
Right.
What else could it be? I mean, it takes up all the different DNA stains. It takes it up, but no one is bold enough to sequence it yet. Although I have colleagues, 3 different creation-based laboratories are now starting to look for. Again, we've had some that looked in the past, about a decade ago. They didn't find any DNA in their dinosaur samples, but now we have 3 labs that are doing it right now, and learning how to use, because a new sequencing technology has come available in the last 2-3 years called nanopore. So Oxford's company makes nanopore sequencer, and so it's a different technology, and it's able to detect sequences that are as short as 20 base pairs long. So that's a lot shorter than the older sequencing technology. So now we have. So in other words, if the DNA has degraded, it used to be, millions of bases long. Well, that gets chopped down, and chemistry happens to those pieces, and the fragments the lengths get shorter and shorter as the degradative chemistry progresses. And so, do we have anything left? Any sequence left? And we may watch this space, because the preliminary results are starting to show that there may be sequences.
Okay.
Yeah.
So what does this mean for our hypothetical Jurassic Park?
Well, okay, if we find dino DNA, okay, according to how old it is, according to the flood, let's see, these are flood fossils, and if it's 4,500 years ago, you would only get, at best, really short, tiny bits of DNA sequence. I don't know, 30 bases long would be a nice. And so it's so far down the chain of DNA degradation, decay is a better word, that you've lost all the information, almost all of it. It would be like you've got an encyclopedia. Let's say it's a 2-volume, double your size of the two shelves worth of one encyclopedia. Let's just imagine that. Back when we had books that we used to look at,
yes,
for encyclopedias. So imagine that is the total set of information in that dead dinosaur DNA. And so, but over time, the books fall off the shelf, and then rats get involved, and they start tearing pages, and insects eat the paper, and then you give it, thousands of years, millennia later, you might find one letter still off of this page, or one half of a word off of that page. Even like we do with archaeological artifacts, even they get damaged in this way. And so, by the time enough time passes since the flood, let's say, 4,500 years passes, then you've got so little information that you could never, piece it together, because you wouldn't know where this letter came from or the context for that half of a word, and where does it go? How does it fit? And what else is missing? Like in the Jurassic Park movie, they knew they would have a lot of missing DNA, so they just filled it in with like frog DNA or something.
Yes.
So, which is again a lot of fairy tale with a magic wand. Let's just frog DNA this, ding,
So maybe we don't have DNA, but we do have biological tissue, correct?
Oh yeah.
And we have a lot of that, from my understanding, more and more. and that is actually your expertise. Can you tell me just like a brief summary of that particular science in just a couple of minutes, and then we're going to talk about some of the responses that maybe the other side would give to finding those things.
Sure. Well, it's pretty straightforward. It's a descriptive science. So we're just describing what's there. And brave mainstream scientists are actually looking for molecular signature. And they've used 35 different independent techniques to describe all kinds of biological materials, organic, even whole tissues. There's blood vessels, branching blood vessels, sometimes blood vessels with red blood cells in them. They found nerve tissue. They found, with like myelin sheathing still on the nerves. And this is all published in the mainstream secular technical literature. And why in the world it hasn't made it down to our regular textbooks, I can only imagine, because it's really solid science. So I've been collecting these reports over some number of years now, over a dozen years, and we've got, I think the list that we curate is up to 130 different published reports of original biomaterials, whether it's molecules, whether it's proteins, or whether it's even whole tissue fragments. It's always if it's whole tissue, it never lasts long once we expose it to the air in the lab. I say we. I've been looking, and I've found fragments under the microscope, but not that you could see with your eyes. But others have found bits small enough that you could see it with your eyes, with unaided. So anyway, but most often we find molecular signatures of all kinds of different proteins.
And this is more than just dinosaurs, correct?
Correct. Yeah, we're talking turtles and birds and all kinds of creatures. So and a lot of these are from, I would call it middle of the flood layers. So what's interesting is we find these tissues in some cases in like worms, sponges that are in the lowest layers that have any fossils. And some of these are called Cambrian. That's the conventional name for these layers. We would tend to think of them more of like the initial flood layers early in that same flood year. And so the Cambrian, with its age assignment of over 500 million years, there's just no way you would have any tissue left. After 1 million, it's unthinkable. But 500 million, it's ultra fiction or something like that. But yet we still find it down there as well, as though the Bible's expectation and our flood model that we've built on that Bible is right. Yeah. That all this whole rock stack with even the layers way down there were not deposited in some eons over across eons, periods and epochs and ages, and all that is a human construct. We think that the rock stack was deposited in large part during just 1 year, 4,500 years ago. So you don't need a lot of time to make thick rock strata if you have a lot of water. And the Bible says there was a lot of water, and it covered all the high hills that existed in that pre-flood world. Those were totally covered. So according to that model, no wonder we have these molecules and tissues, histone, elastin, laminin, collagen, ovalbumin, hemoglobin, and many others have been published in the literature.
That honestly sounded like you were listing a series of pharmaceuticals. But yes, a lot of different pieces from a lot of different creatures. So that begs the question, what do others say about that? If we know that these supposedly Cambrian from that layer, if we know that that can't exist, what's the reasoning that evolutionists or uniformitarians would give for those still existing? I know that I've heard bits and pieces about maybe like iron preserved it or something like that. But what are some of those arguments that are made, And what is your response to that?
Well, honestly, I think one of the more common arguments should be an embarrassment. And it is for them to say, oh, that's just bad science. You didn't actually find what you've just described here. They just simply deny it. And it kind of reminds me of the Pharisees denying that Jesus was God because it just did not compute. They refused to calculate that the possibility that this guy could actually be the Messiah in human flesh. And he proved and proved and proved and offered every proof possible. He even raised people from the dead in front of them. And they were like, well, you're doing these miracles by the devil is what they accused him of. And so it's that kind of abject, shameful, shameless denial, So they're just denying it. Once you get past the people who just deny it for no good reason, no scientific reason anyways, then we get some of them who say maybe there's a way to preserve it. And actually the iron preservation hypothesis is out there. It got published as a possibility. You have iron that comes from the blood and other tissues, and it facilitates this kind of chemistry that does this, that, and the other, and turns some of the proteins into something like plastic, which might last a lot longer than the actual original proteins. So that's the story. No one has tested that story, and we're interested in actually testing it. And we have labs working on that right now. So, but meanwhile, what do I have to say about that? Well, if it's untested, then it's at least premature to say this is a valid rescuing device. Because of iron, therefore, we can now assert that these proteins are 80 million, 90 million plus years old. No. You didn't test. That's not a scientific conclusion. It's just a rescuing device that comes from our imagination until it gets tested. So that's what I think about it. Plus, I have other thoughts about it, and I've published these in all kinds of articles on ICR.org, so we can go in greater depth for people who want to read about it. But the chemistry that they're proposing is actually more destructive than it is constructive. So every reaction that it might do to build proteins into some sort of a plastic, it 1,000 times more often, it will take those proteins and break it down even faster than they ordinarily would decay. So iron is no help.
Right.
Iron is no help. There was a paper published where they did test iron on fresh bone, and they found that in iron solution, it actually accelerated the decay of the collagen that made up that. I think it was a chicken bone, and I can show you that paper in the lower third. So it's not a rigorous test, but it's an initial way to test the iron hypothesis, and boy did it fail that initial test.
But yet it's still being.
But yet it's still pushed. In fact, it made it into Jurassic World, the 2015 one.
There's too many of them.
There's too many of them. Yeah, it's the kid's name is Gray, and they're in that bubble car thing.
Yes.
And the little kid's talking to his bigger brother, and he goes on this mini soliloquy about iron preservation, and iron can preserve the DNA. So and it was like the writers putting this in. Hey, this just came out in the literature about iron preservation. Let's throw it in the movie, and that will legitimize the whole fiction of getting DNA out of dinosaur bone and then building Jurassic Parks out of that. So I think it's more fiction.
Yes.
That's what I think.
More and more fiction. Well, speaking of more fiction, in the first Jurassic Park, one of the main plot points is that scientists had to kind of like cobble together some of the DNA. And so they filled in the gaps with frog DNA specifically, and that caused some issues narratively, of course, but I'm more concerned about the science of it. Is there technology that allows for the splicing together of two separate DNAs?
Oh, sure.
Yeah.
Yeah, you can splice DNAs, but what we just talked about with the encyclopedia that you need. Well, let's compare it with modern cloning. And so every few years, someone comes up and clones a different kind of animal. So they've cloned, of course, Dolly the sheep was the first. But there's companies now that will clone your pet. They'll clone your pet cat and dog, and some of the clones that they've been able to. These are whole animal clones. If I say the word cloning, when scientist says the word clone, they're usually talking about just cloning a single gene or a segment of DNA. And that's really, really common, everyday laboratory stuff. In fact, I did cloning for my biotechnology degree, and then back in the 1990s. Ooh. But I didn't clone whole animals.
Right.
That's rare. We just cloned genes. That way you can mass produce a protein, and then you can purify the protein and test to see how it behaves, to learn how that protein works, what that protein does, how that protein behaves with other proteins, and usually with the mind of, okay, now that we know how it works, we know how to treat diseases. So it's usually health related. But in this context, we're talking about whole animal cloning. But that's become, but what are we doing with these whole animal clones? We already have mommies. We already have a female cat into which we can place this fertilized egg, artificially fertilized egg. And then, so the womb takes care of the rest. So that's how it goes in placental mammals that we've cloned. But you already have the cell is already there, the mother cell, and the womb that nurses and takes care of, and the placenta, which forms this symbiotic. You could maybe by analogy call it like they help each other. And the placenta actually becomes a slave to the developing baby, and baby tells mama, okay, I need some of this, and mom's body supplies it. So how are you going to replicate that entire process that goes on in a womb? I'm not saying it's impossible. I'm saying it would be an enormous technical hurdle to try to, and that's if you have an egg. Mm-hmm. Like if you don't have the egg into which to put your DNA, where do you even start? I mean, you'd have to start with some kind of egg of some kind of animal. But the thing is, that another technical hurdle would be, it turns out that the machinery that handles DNA, the machinery that takes the correct volume off the shelf, opens it to the right page, reads the right instruction code, so to speak, and then knows what to do with that instruction code. All that is specific to each created kind. Mm-hmm. So if you take, DNA from one kind, stick it into a cell of another kind, well, that cell's machinery is going to go, where's the volumes that I know about? This is a different instruction set, and it's not going to know where to start. You're not going to get a clone. We've tried that already. It's just the cell even stops dividing. It doesn't even start dividing, or it may divide to 84 cell division or something like that, or 100 and something. But then it's like, yeah, this isn't working out. It's so cell division stops, and you never go beyond and never go beyond that. So
it's fascinating, and
there's a lot
kind of weird.
There's a lot of biology,
yeah.
And the more we learn about this biology, the more details we discover, and each detail it's like a technical, hurdle has been solved by the Lord Jesus when he put this in place and put that in place. And the further down you drill, the more little tools and systems are in place to make everything go and make everything happen for each created kind. So when they have these vials and these coolers on the movie, and then they have a guy with a lab coat, and it looks like he's really doing science. Not at all. I mean, it's complete fiction. In fact, it's more like magic, pulling a rabbit out of a hat, pulling a dinosaur out of a cooler, Yeah. It's a trick. It's not, there's nothing real, in terms, you're not actually the magician, or whatever we have to call them now. Illusionist. The illusionist is really doing an illusion. He's not actually taking molecules, and building a rabbit out of thin air and putting it into the hat. Like Jesus can do that. In fact, he did that. That's how we got here. It's the only way we could have gotten here is by a big giant miracle called creation. And no wonder we're so complicated in our structure. So he can do that, but we don't have the tools and intelligence and capability to construct systems that are this small, this precise, and this overwhelmingly complicated.
That reminds me of just not super recently, but kind of recently. It was in the news. They're like, "Oh, we brought back dire wolves." Do you remember that? Do you recall that being in the news? And I read a lot about it on a very surface level, right? But it was, some people were like, "Oh, these are real dire wolves." Other people were like, "No, they're like, they used some dire wolf DNA and some living dog or living current wolf DNA." Is that, do you know anything about that situation?
Honestly, I don't know much. I kind of read the headlines like you did on that one, but I think what they may have done is take dire wolf DNA sequence, specifically the sequence that controls, I think it was hair or something, the fur or maybe body size. Anyway, there's a specific region of the DNA that they know that it plays a role in some aspect or feature that was dire wolf specific to dire wolves, and then they patched that in using new technology, which is now available. Like CRISPR-Cas9 is the famous one. It came online, a decade ago, and now in only 11 years, it has gone from research and development to through clinical trials, and in 2023 was approved to be used on humans to treat like sickle cell anemia. And what it does is it goes in. It's a system that bacteria already had that scans the interior of their cell for DNA that it might want to get rid of. So by combining that system with some other molecular tools, they're able to put these little they're like nanobots, really. I mean, kind of, but they're able to put these into human cells, and then what it does is it finds the region of interest. It finds the region in your DNA where you have that, you've inherited that mutation, and it's a single base, change that has taken place in one of your ancestors. It's so it finds that sequence because you tell it which sequence to find now, and then it cuts the DNA right there, and so then it facilitates it recruits a DNA repair mechanism that's already in your cells to go ahead and patch it up correctly, and it tells the sequence, here's the correct base. So it's helping. So you can help people with genetic diseases like that with this technology, and it's amazing. It's amazing, and so I imagine that you could use that similar technology to take and splice into a genome, into an egg cell, and build it up, and then put that egg cell into a live wolf. There again, you got the mama available and ready,
right,
and still alive and not extinct, and put that in there, and so you can call it a dire wolf. And its DNA is it's probably more similar to the DNA that dire wolves had. It must be because they've copied the sequence.
Right. But
I don't think they have enough of the dire wolf sequence in those frozen bones from which I assume they got it to even build out a whole dog. But they have enough of the sequence to where they can piece it together and go, maybe these were the DNA differences. So the other thing about that is, if I can just add this. It's not just about the DNA. We're so DNA focused, but it's about all those machines that are inside the cells that are deciding which volumes to pull off the shelf. Which sequence of DNA do I unpack and read and deploy and transcribe and make transcripts out? And when should I do it? Should I do it early in development, late in development? Which tissue should I do it in? Should I do it in brain tissue, muscle tissue? And so that's why every one of your cells in your body has the same DNA. But you have all kinds of different cells.
Right.
Well, but they have the same DNA. Well, what's the difference? Well, because these cells are, as they differentiate, they're pulling different volumes off the shelf. They're accessing different pieces of information from that same encyclopedia. So, what was the dire wolf? It probably had something very similar to a regular wolf, but do we still have the same tools, the same RNA sequences, the same histone lockdown codes? This is called epigenetics. I'm probably using too many big terms for a podcast, but anyway, yeah, there's systems that actually close the volume. Like, you don't need this volume of this, or you don't need pages 500 through 612. So let's put a big paper clip on that. So these paper clips on your DNA are critical, to the outcome of what a creature is going to, the specific variations of each trait that a creature is going to have. So not only do you need the DNA, you also need these other codes there's very little information on what those codes might have been for extinct varieties. And remember, this is a variety of the dog kind, the dire wolf. So it's not like if we bring it back that we've done a lot. We've just altered a dog.
Yeah. Wow.
That was probably TMI.
No, I love it.
I hope you can edit that down to something useful.
No, we're going to leave that all in there. No, we'll probably trim it down a little bit, but okay. Awesome. Thank you, though. I know that was kind of off the cuff, but okay. So it kind of all boils down to Jurassic Park, as we see in the movie or in the movies, so many of them now. It's not possible. It's fiction. That's what I'm getting from our conversation.
If it's not possible, it's right next to impossible.
Right. Okay.
So, and people say, well, you would have said it was impossible to do gene editing, but now we can do that with CRISPR-Cas9. And I have to grant that.
Sure.
So back then, there are certain things or it's impossible to sequence DNA. Well, now, if the DNA fragments have gone down to where they're only 20 bases long, well, now there's new technology that can do that. So now we're back on the hunt to see. So there are cases where we used to think it was impossible with that technology. And so I'm not going to say it's totally impossible, because maybe someone can build a giant womb. I mean, I say giant because the things we tend to build tend to be way bigger than the way the Lord Jesus built it.
Right.
Maybe someone someday can, piece together all these little pieces of a cell and build something from scratch. But the technical hurdles are just absolutely overwhelming, and it looks impossible from where I'm standing.
Right. So we can't have a Jurassic Park today, but they were real creatures. We have the fossils.
Like this one here.
That one right there. But what was the world that they lived in like?
Yeah, I guess I would say this is a good opportunity to just get a picture, a snapshot of dinosaurs through world history, and to get that from the Bible, starting from the Bible. So if we do that, then we say they were created, or we could say formed, and that's Genesis chapter 1. God created all the great beasts of the earth, and I think that's what happened. And so they would have been land-dwelling creatures created on day 6. But then it wasn't long. I mean, it wasn't long at all, I don't think, until the fall happened into sin, and therefore the curse on sin. And it was a death curse, really. And so now Romans 8 says, all creation is groaning and travailing together until now. And it was subjected to frustration, but in the hope that it would be redeemed. So now we've got this giant story arc into which we plug these dinosaurs, created, formed, and then fallen, we could say, really soon. And then the third event is 1,656 years after creation and the fall. We get that number from Genesis 5, the chronology that's in there. We have the flood. So Noah's flood was world destroying. All that water came from great depth up through the crust, rearranged the world. Now we have new continents, And then those waters ran off into today's oceans, which are now deeper, I think, than the pre-flood oceans. So flooded. And during that flood, these creatures got buried alongside birds and turtles and all kinds of other creatures, mostly swamp creatures, I think, or wetlands type creatures and plants as well. And then so the high ground living creatures got buried last because it took month after month for the floodwaters to reach the high and dry places on Earth where camels and people would have been. And so there's not as many fossils of those available to us anyways. And then, but then since they had, what does it say in Genesis chapter 7 about the animals that God brought to Noah? And then a later verse says, and Noah brought the animals onto the ark. So there's a partnership between them, God and Noah. And what animals ended up on the ark? The ones that walked on land, because you didn't need to bring fish on the ark. They're outside the ark, and most of them died, but some of them survived. And that's how we get the ocean and lake creatures that we have today. But it says that had the breath of life in their nostrils. And so what do we see in their skulls from these dinosaurs? They had nostrils and legs. So they probably were on the ark. I say probably because I wasn't there. I don't know. Maybe they could have survived like crocodiles, because amphibious creatures were kind of like, do you need them on the ark? So there's a question mark on that. But we've been saying for decades that dinosaurs were on the ark, and they certainly could have fit, no problem. Even these sauropods like we've got here. By the way, I've had this since like the 1970s. This is my brachiosaur toy, and it was featured in Jurassic Park 1993.
The first one, yeah.
Incorrectly, but I thought, I'll just bring it as a homage. So even those would have gone on the ark, but they don't have to be the giant grandparent versions. They're just the juvenile ones. The smaller ones the younger ones would have fit. So formed, fallen, that's what I said, and then flooded. And then after the flood, they might have survived. That's why we have legends of dragons, I think some of them, and are legit. And that's, I think, why we have Behemoth in Job is one of those true dragon legends. And I think it was talking about a sauropod dinosaur there in Job. So, but after the Ice Age, we have these massive climate changes that are, long-term results of the flood on Earth. And so then their habitats dry up, and now you've got faded from memory. It faded from existence because one by one they die until many of them eventually went totally extinct. And maybe they're all extinct by now. So formed, fallen, flooded, faded, and then found. We could say that they're found by paleontologists.
Like you.
As evidence of the flood that was really happened.
That's excellent. And I'm kind of glad that we don't have to worry about them wandering about today. Again, we have 6, almost 7. By the time this podcast airs, we'll have 7 Jurassic Park / World movies telling us why living alongside dinosaurs today is a horrible idea. So maybe it's for the best. But do you have any concluding thoughts? Anything you want to share with our audience before we close it out?
I want to just say a few words about cloning because it's pretty controversial. And
sure.
There's a lot of ignorance. And not that I know a whole lot, but
sure.
just a little about it. Cloning is here, and there's even whole animal cloning. And although we don't have cloning of dinosaurs, and I don't think we can clone something out of extinction, because it takes a mama to make a baby, as far as current technology. But we do have some forms of cloning. And it's a powerful tool, especially when we're talking about cloning just individual genes. We could clone them up. Just makes copies is all it means, and put them into different. So we most of what we eat, has some cloned. It's transgenic. We take a gene from over there and stick it in here, and then grow it up in a plant. And anyway, it's a powerful tool. It's a powerful tool, and it's so powerful that we have to be careful about how we wield it. It's like a car. You can use a powerful tool, a car, to get from point A to point B, but you can also use your car as a weapon, like these terrorists sometimes do. So it's a powerful tool. So use it for good. Use cloning for good. And then it also, I would say, it ties into the it's a tool that could be used positively in order to do the dominion mandate. So we talk about the dominion mandate, Genesis 1, I think it's 28. And God blessed them, and God said to them, be fruitful and multiply, fill the earth and subdue it, and have dominion over the fish of the sea, the birds of the air, and over every living thing that is upon the earth, that lives upon the earth. And so we have this dominion mandate, and it's never been rescinded. Mm-hmm. So God's intention for us humans is to take care of this place. So we have to understand how it works. And as we do that, as we learn about how intricate and complicated his world is, and especially the world inside of a cell, it should cause us to worship even more. We should say, wow, I never knew you were this smart, God. You are so smart, beyond what I could have even imagined. And then also, thank you, Lord, for giving us these tools. Help us to use these tools for good, to take care of your planet that you've provided for us as a part of the dominion mandate. So I think we have a biblical basis for doing good science, descriptive science, for doing good, for developing new tools that can help people, help rescue people from diseases even. I think those are good uses of these kinds of technologies that we need to thank the Lord for as well.
Absolutely. And I just wanted to say, as it came to mind, you had mentioned, the scientists who just deny the soft tissues that have been found. You likened them to the Pharisees. You said they didn't believe, but some of them did believe.
Yes, good point.
Some of them did. And so, we can hope and pray that these discoveries might cause some of these scientists to rethink the narrative that they've been told about world history, the universe's history, and maybe the Holy Spirit will work in their hearts in that.
I mean, regard. That is the logical, if you just go with the science, we have these proteins, we have at least chromosomes, mm-hmm. and they should not be there. Absolutely should not be there. If it's nearly any fraction as old as the conventional, age assignments demand. And so what do you do? So that's what I've done is I've gotten rid of the conventional ages from my imagination, mm-hmm. and I've replaced them with the Bible's timeline. And that's okay, because it's consistent with the scientific evidence. But it also leads me to belief in the Bible, which leads me to belief in the God of the Bible. I think that's a side benefit as well. So yeah, let's pray that someone will follow this evidence where it leads, and even as some have done.
Yeah, absolutely. Awesome. Well, thank you so much for joining me today on this podcast. It was a blast to chat with you, and we'll have you back on again soon. It's always a pleasure. So thank you.
Thank you, Trey.
Absolutely. And thank you to all of our viewers and listeners for joining us on this podcast today. Make sure to like, subscribe, share, get the creation message out. And actually, I want to draw your attention to these wonderful members and patrons. Their names are scrolling down the screen right now. These are people who have decided to take the next step and join alongside us. And because of that, they get several perks, including getting the Creation Podcast a week early and a couple of other perks, depending on which tier they're at. So if you want to become a member or a patron, you can click on the links in the description below. And in honor of them, we do have a question for Dr. Thomas, a little spur of the moment question. Are you ready?
Bring it.
Okay. This is from David Chu. Okay, that's a real name. Are Genesis 1 and 2 parallel stories?
Well, so Genesis 1 is the creation account, but then Genesis 2 zooms in and retells the creation of Adam account. And that's kind of more like from his perspective, from what he would have, from what Adam himself would have experienced and known. So anyway, they emphasize different aspects, but none of those aspects contradict one another. So they complement one another. So they're parallel in that sense. And in fact, the Lord Jesus treated them both as equally historical. And he quoted them both in Mark 10, verse, I think, 7. So in verse 6, he says, haven't you read, he that made them in the beginning made them male and female. Then he says, and the two shall become one, which he got that from chapter 2. Male and female, he got that from chapter 1. So he's directly quoting snippets from chapters 1 and 2 seamlessly in one sentence. So if these contradict one another, which they don't, or if these indicate that the histories were scrambled together and cobbled together, like so many Bible skeptics want to say, then we have to say as well that Jesus, having not treated them that way and having believed them and used them as the basis for his argument about marriage, the history of what happened is the reason why we have marriage and why we have marriage the way it is what he's saying to the Pharisees there in Mark 10. So if we're going to scoff at that, we'd also have to scoff at the Lord Jesus, which is of course also what they want to do,
right? Because
we don't want him being the boss over me. But the facts remain 1 and 2 complement one another. I've got an article on that on icr.org. You can just type in keywords in our search engine and find that article. And we have other authors who have written on that as well over the many decades that we've been around. And by the way, thank you for all the support from our members and patrons.
Thank you so much, Dr. Thomas.
My pleasure.
Absolutely. And thank you again so much for being here. We'll see you next time on the Creation Podcast.